Welcome to your last lecture in 2014, section 10.4, protein synthesis. Let's talk about the flow of information, which will also review some of your cell organelles for your final. We know that DNA, which resides in the nucleus, contains the instructions on how to build a protein. So when we make a copy of our DNA, what we're going to turn that into is we're going to take DNA and we're going to make what's called messenger RNA. The messenger RNA, so if we're in here, we make our copy. The messenger RNA is then going to go out one of the nuclear pores and then find one of the ribosomes. And remember, ribosomes are the organelles that make proteins, and it's going to translate that mRNA to a protein. And we're going to talk about these processes in this section. Last section we talked about taking DNA to DNA, and that was called replication. In this last section we're going to talk about transcription and translation. There are three types of RNA. So we have what's called mRNA, which is from transcription. We're going to talk about that. And that's going to tell the ribosome how to assemble the protein. And then we also have tRNA, which got cut off here. And that is going to be responsible for bringing the amino acids to the ribosome. And then we have rRNA, which is responsible for actually making the ribosome. All right, so we have a little problem with the formatting. So transcription, let's talk about that, although it's kind of all messed up in the corner of the slide, but it should be fine on your student copy. So what we're going to start off with is we're going to start off with our DNA, and in replication we saw we went from DNA to DNA, but in this case we're going to unzip. So we unzip our DNA. So we open up those, we break those hydrogen bonds, and instead of DNA polymerase coming in and making a copy, we're going to have RNA polymerase coming in. So our RNA polymerase is going to read down the DNA molecule in complementary base pair in order to make our messenger RNA. So what is a codon, or we can even just talk about a start codon? So first of all, what the codons are is we're going to read our nucleotide bases 3 at a time. So those 3 bases are referred to as a codon. Now the start codon, again, is going to always be AUG, and that's what's going to tell our ribosome where to start reading our genetic code 3 at a time to build our protein. We have to have some point, a reference point as to where to exactly start reading this message. So let's talk about the ribosome. Now, we had said that the ribosome is made up of rRNA and some proteins, but what's going to happen is our ribosome is actually in 2 pieces. And the first thing that will happen is that bottom piece will snap on to the messenger RNA at the start codon. Now the start codon is always going to be AUG, which codes for methionine, so MET. So here is our start codon, or actually you could say the pocket that contains our start codon, and it will, the tRNAs are going to pop in and out until one actually will complementary base pair with that codon, and once it does, then it knows that it's a proper fit, and then the ribosome will move over. So once it moves over, then what's going to happen is, as we notice here, we have our string of amino acids. So our amino acids right here, 1, 2, 3, and 4. So right here's our 4 amino acids. They're going to move over from one tRNA to the next. All right, and then we're going to move down so we can get to the next codon to read. So this is a chart of the genetic code. Now, in order to read the genetic code, we're going to have to read it first letter, second letter, third letter. We're going to practice that today with our connects. But let's just say, for example, if our first codon is, like we said, A-U-G, how we're going to read this is we're going to look at the first letter, which is A. So, and then we're going to look for U. So we know if we go down and across, we're in this quadrant here. For the third basin, you're going to go over to the far right, and you're going to look up that G. So you're going to go all the way across, and that amino acid that we get is M-E-T. If we were to, let's see, let's do that again. Let's do another one. Let's just pick C-G-U. So we'll go to the first letter, which is C, and then the second one, which is G. And those two intersect at this box. And then we're going to find our third letter, which is going to be over here. It's U, so we're at U. So here's our CGU. So that next amino acid is going to be arginine. All right, so we're going to practice using this genetic code today. You need to be able to do this. This process is called translation. So as the ribosome is sitting on the messenger RNA and we are reading our codons, whoops, here's our first codon, here's the second codon, the third, we keep going. All right, so as we have gone through, we can see that the tRNAs have complementary base pair. This right here is referred to as the anticodon, because it's the opposite. So the anticodon is on the bottom of the tRNA. They will then join together right here, those amino acids, and form, you should know, that will be your peptide bond. Once that peptide bond is formed, then everybody's going to scoot over, and this one's actually going to be released, and then a new tRNA will come in and try to match up with UCG. And they'll continue to do this as this polypeptide chain grows. That polypeptide is going to be our protein that we're making. So let's just go over translation. So you have your DNA, and you unzip it right here. You make your messenger RNA. The messenger RNA then leaves the nucleus, goes into the cytoplasm, where the ribosome will pick it up, and then it reads it. The tRNAs will bring the amino acids, and as it's reading the codons, it will assemble the polypeptide, which will then make your protein. So again, translation is taking your mRNA and converting it to the actual protein. Now, on the bottom of your tRNAs, remember that's your anticodon, and the three that you're reading are called the codon. And then again, of course, you've got your amino acids there. So they're just going to keep coming in, popping out, keep going in, popping out. And the ribosome's going to keep reading along the messenger RNA. So we're going to keep reading along the messenger RNA until we get to this release factor, and that is going to be a specific codon as well. There's one of the stop codons, UAA. There are 3 stop codons. So we only had 1 start, but we have 3 stop codons. When it reaches that stop codon, when the polypeptide goes to form that peptide bond, there's not another amino acid for it to join onto. So it will release the polypeptide chain, which then has the ribosome disengaging or falling off the messenger RNA, and translation is done. So to review, the first thing we did in this chapter was we talked about DNA and how we can make a copy of DNA. So DNA to DNA was called semi-conservative replication. And then we went from DNA to messenger RNA, which is called transcription. And then from the messenger RNA, we went to the ribosome and we made our protein, and that was called translation. So that concludes the lessons on chapter 10, and that is your last lecture of 2014, at least for biology.